This is the Professor, CBMD PhD, and today's Monday, November 25th, 2024. It is 8:48 and 16 seconds, p.m. Eastern Standard Time, and today's research, micropinocytosis and macropinocytosis. So let's hop to it here. Micropinocytosis is a non-specific ingestion of fluid and small protein molecules via small vessels, usually smaller than 150 nanometers in diameter. Micropinocytosis is performed by virtually every cell in the organism, and it is constitutive. An important example, it involves a continuous dynamic formation of small vessels at the cell surface. The vessel formation of micropinocytosis is usually associated with the presence of caveolin and flotilin proteins that are found in lipid wraps. Caveolin 1 and caveolin 2 are found in all non-muscle cells, except neurons and white blood cells, whereas caveolin 3 is muscle, cell-specific. Flotilin 1 and flotilin 2 are found in vessels distinct from caveoli. Also mechanoenzymes such as dynamine, GTPase, family of membrane scission proteins are involved in pinocytic vessel scission, the process of pinching off from the plasma membrane. Pinocytic vessels are visible with the TEM, and they have a smooth surface. They are especially numerous in the endothelium of blood vessels and in smooth muscle cells. Because caveolin 1 forms complexes of 14 to 16 monomers that effect changes in membrane curvature leading to vessel formation, micropinocytosis does not require clathrin. Micropinocytosis also does not require remodeling of the actin cytoskeleton and therefore may be referred to as clathrin-independent and actin-independent endocytosis. Macropinocytosis represents a non-specific uptake mechanism for extracellular fluids, solute nutrients, and antigens. In this actin-dependent process, the actin cytoskeleton is rearranged at the plasma membrane, leading to formation of surface membrane ruffles. The membrane ruffles becomes elongated and then fold back onto the plasma membrane to entrap extracellular fluid. They give rise to large 0.2 microns in diameter endocytic vacuoles called macropinosomes. The large fluid-carrying capacity of macropinosomes is utilized by cells of the immune system, e.g. macrophages and dendritic cells, in order to sample as much of their extracellular environment as possible. The amount of solutes and membranes internalized during macropinocytosis exceeds that of any other endocytic pathways. Macropinocytosis is a regulated process that occurs in response to various growth factors, such as macrophage colony stimulating factor 1, CSF-1, endothermal growth factor, EGF, or platelet-derived growth factor, PDGF. The macropinocytosis undergoes a defined sequence of maturation steps in which their contents are either degraded in the late endosome or lysosome or recycled back to the plasma membrane. Because of the initial increase in actin cytoskeleton remodeling in distinct regions of the cell surface leading to formation of plasma membrane ruffles, macropinocytosis is referred to as clathrin-independent but actin-dependent endocytosis. And this is Professor Christopher, codename Will, leader of research scientist ufologist, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover, passion, world peace. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day. This is the Professor, CBMD PhD, and today's Monday, November 25th,